Anatomy of stem in Pinus. Coming to the long shoot, secondary growth is similar to that of a dicotyledonous stem, and the cork cambium cuts off cork towards outside and few layers of secondary cortex towards inner side. Many tannin-filled cells and resin canal cells are distributed in the primary cortex. The cambium cuts secondary phloem towards outside and secondary xylem towards inner side. The primary phloem is crushed and pushed towards outside by secondary phloem. In the secondary xylem, annual rings of thin-walled spring wood formed during spring season and thick-walled autumn wood formed during autumn season are present alternately. Such a compact wood is called as pycnoxylic, and the age of the plant can be calculated by counting the number of these annual rings. Below the secondary xylem are present a group of entire primary xylem. Some of the medullary rays connect the pith with the cortex and are called as primary medullary rays, while the others run in between secondary xylem and secondary phloem are called as secondary medullary rays. The central part of the stem is filled with parenchymatous pith. Resin canals are also present in cortex, secondary xylem, primary xylem, and rarely in pith. Coming to the tangential longitudinal section of wood. In this, bordered piths and medullary rays are present in sectional view. Each bordered pith is enclosed by a pith chamber bounded by a pith membrane and contains a centrally located solen torus. Tracheids are composed of rectangular cells. Middle lamella is very clear. Many uniseriate medullary rays are present. In the xylem region, medullary rays contain a centrally located starch cell surrounded by tracheidal cells. Albuminous cells are also present in the medullary rays in the phloem region, and pith is absent. Coming to the radial longitudinal section of wood or secondary xylem. In RLS, the stem is cut along the radius, so the pith is visible. It is bounded by externally by cortex, cork cambium, secondary phloem, and crushed primary phloem. Bordered piths surrounded by bars of sinew in tracheids are seen in surface view. Uniseriate medullary rays run horizontally. In xylem region, thick medullary ray cells are surrounded by ray tracheids. Fifth point is thin-walled ray parenchyma is also present, and the xylem is separated from phloem with the help of cambium. Albuminous cells are present in the medullary rays in the phloem region. The phloem consists of C tubes, C plates, phloem parenchyma, and pith is present. Next is anatomy of dwarf shoot. So, the transverse section of dwarf shoot, that is young dwarf shoot, is similar to that of young long shoot except the following differences. The number of resin canal cells present in the cortex is 6. The number of vascular bundles is also 6. Pith in the dwarf shoot is comparatively smaller than the long shoot, and the structure of vascular bundles are conjoined, collateral, open, and endarc. Coming to the old dwarf shoot, in a transverse section, even this is similar to that of long shoot in many aspects. Cork cambium, and secondary cortex are not normally present, but the epidermis is surrounded externally by scaly leaves and followed internally by multi-layered cortex. Inner to cortex is crushed primary phloem, secondary phloem, cambium, and secondary xylem with medullary rays. Protoxylem is indark. A small pith with some tannin cells is present in the center. Thank you.